In last module, I'll be talking about the drugs from marine sources which are presently in market or in pipeline. So here is the current status of marine derived drugs. Over 15,000 different marine nature products have been described and hundreds of patents have been filed so far. More than 20 different marine nature products are currently in clinical trials. In my previous module, I have already explained there are 4 different clinical trials, and any molecule which we want to develop as a drug should pass through all 4 clinical trials. So mostly time consuming and very rigid process of clinical trial. And more than 5 different marine nature products are currently in market. In 1975, Era A, first drug in medical use from marine sources, was launched. That was in 1975. Here you can see the reference of this. And after that, there are many other nature products are launched in market as a drug, and some of the molecules or nature products are in clinical trial. I would like to give you an example of molecules which were discovered or which are discovered from the sponges. In my previous modules, I was stressing about the sponges. That was the first animal phylum, that is Porifera. These organisms are from Porifera, and these sessile organisms are very potential for marine nature product drug discovery. So from sponges, if you see, that was a first molecule, Ara A, citarabin, Ara C, Ara A, then some more molecules from the sponge which are already in market. Couple of molecules are in stage 2 clinical trials, and some molecules in phase 1 clinical trial, some molecules are in discovery stage. So we will see in this slide which are the products which are currently in market. Okay, Era A, I have already explained you about. This is from the sponge. The biomedical use is anti-cancer, that is leukemia and lymphoma. Then peptides from polar fish. These are antifreeze glycoproteins. These proteins are having role in nutraceuticals, especially for cell protection during cold storage and improved quality of frozen foods. Then chitosan from crustacean shells. Chitosan is useful in cosmetics and wound dressings. And finally, proteins, that is a green fluorescent proteins from jellyfish, the biotechnology product for reporter gene. So these are the some of the products I have explained you here. Also there are omega-3 fatty acids from marine sources, from especially from marine fish for heart diseases. So many other products are there in cosmetics. Also some of the products from seaweeds, for examples, are good or in use in cosmetic industries. So these are the products which are in clinical trial, we have seen already, and the products which are in market. So now in this particular module, I will be quoting some of the interesting stories or the molecules which are in clinical trials. For example, plitidepsin. This is an anti-cancer molecule. Okay, you can see the molecular weight and molecular formula of this. The original or the source of this molecule was Alpidium albicans. This is a tunicate or acidian. So originally this molecule was isolated from the acidian. Of course now molecule is in clinical trial 3. It is a final almost final clinical trial, but now molecule is no more available from Alpidium from acidian, because as per my previous saying, I was mentioning that we cannot destroy animals in large quantity. So molecule is being produced by synthetic route now, and which is in clinical trial 2. Here is the acidian from where the molecule was isolated originally, and is the structure of this plitidepsin, the anti-cancer molecule which is in clinical trial. Then Karen 7000, this is again anti-tumor molecule. Okay, see any, especially in case of anti-cancer molecules, there are different targets. Okay, so by using these different targets, molecules are being discovered and used in the patient. For example, cytotoxicity is a very basic assay where we can see that we can put cancer cells in the multivales, and we can put the drugs or extract or molecules, whatever, okay, and check after 24 hours, 48 hours what is happening, whether those cancer cells are getting killed by this molecule. That is a simple assay. But after that, there are couple of different targets in anti-cancer drug development. That is, for example, tubulin binding or microtubule interfering agents. So what is happening? Tubulin is a protein which is present in, say, cilia or in flagella, and also present during mitosis in spindle fibers, where when there are division of the cells, then this material, nuclear material, to two polar ends, the spindle fibers will take. Okay, so there is a presence of this tubulin in the spindle fibers. So what is happening? This tubulin is always in the stage of polymerization and depolymerization. So if you are interfering in between, suppose you are stopping polymerization, then whole assembly will collapse, so that there will not be a mitosis or the cell division takes place. So this is a target. If you want to arrest the growth of cancer cells, you can interfere in microtubule, okay, by inhibiting this tubulin polymerization or depolymerization. Second one is, for example, anti-angiogenesis. Generally, angiogenesis means formation of new blood vessels. So generally, angiogenesis takes place during wound healing or during embryonic development or during tumor growth. Suppose any tumor is growing in your body, okay, that tumor is secreting pro-angiogenic factors towards the blood vessels, because originally there won't be a blood supply to the tumor, and tumor desperately needs blood supply, so that tumor will get nutrients, okay, and tumor can grow. Okay, so the cells which are present in the tumor will secrete pro-angiogenic factor to get a supply from the existing blood vessels to the tumor cells, and this process is angiogenesis, and then tumor will grow. So there is a concept, suppose if we stop this angiogenesis or blood vessel formation when the tumor is growing, then tumor will be starved. Tumor will not get any more nutrition or the blood supply. So those, some of the molecules people are targeting for this angiogenesis or to stop the angiogenesis, that is anti-angiogenesis. So Chiron 7000 is an anti-tumor agent, okay, and it is also showing immunostimulatory activity. This is in phase 1 clinical trial, okay, and it was isolated originally from the sponge. Of course, this molecule is also is available through synthetic route now. Then discodermolide, this is microtubule interfering agent. This was from the sponge. It's a Discodermia desulata. This is a tubulin interactive agent. I have already mentioned you what is meaning by tubulin or microtubule interfering agent. This molecule is in phase 1 and phase 2 clinical trial. Actually, this was isolated from the deep sea sponge, and this discovery was happened in Harbor Branch Oceanographic Institute in USA by Professor Gunasekhara, and his team discovered this molecule. Of course, this is very promising molecules, which is we hope that this molecule will reach in market in the form of drug very soon. So IRA-M, this molecule is already in market. I have already explained you. This is from the sponge. Then manolide. Manolide is anti-inflammatory agent. See, so far throughout my lecture, I was stressing more on the molecules in the form of antibiotics to treat infectious diseases, and molecules for anti-cancer purpose to treat different cancers or tumors. Because natural role, if you see, of these molecules is either to kill pathogens when they are filter feeding, okay, by the organisms. They produce these molecules to kill pathogens which are entering through filter feeding process in their body, or to stop the growth of neighboring organisms which are very aggressive organisms which are growing close to the host species. So they produce these molecules so that cells, rapidly dividing cells of their neighbors, can be killed. So that's why mostly from marine sources, the molecules are discovered in larger quantity as in the form of antibiotics or anti-cancer agents. But apart from that, there are some toxins, for example, which can be used in anti-inflammatory purpose. For example, conotoxins. Conotoxins are very promising in these areas. So here is an example of manolite. Manolite is an anti-inflammatory agent. This was isolated from Pacific sponge, of course, porphyrin, anti-inflammatory agent, also analgesic. This, unfortunately, this molecule was withdrawn from phase 2 clinical trial. It was very successful till phase 1 clinical trial, but after that, the clinical trial people found that there are some problems in clinical trial 2, and this molecule was withdrawn from clinical trial. This molecule, that is LEC-depsin, is a form kalalide group of molecules. It is a cytotoxic agent, of course, and this is from nudibranch or Elysia species. This molecule is good as a cytotoxic molecule, and also it is a gene inhibitor. Okay, currently phase 2 clinical trial. Here you can see a complicated structure of kalalide F, this molecule, and the producing species, that is Elysia rufescens. Then we'll come to a bryostatin 1. When I was explaining biodiversity, I had shown you a picture of bryozoans. Bryozoans are organisms which can find in intertidal regions or subtidal regions. Okay, and this molecule, bryostatin 1, is immunosuppressive agent, and this is from Bugula neretina. This is the bryozoan species, okay, which is producing this molecule, and here you can see the structure of this bryostatin 1. Fortunately, this molecule is in phase 2 clinical trial, and surely it will enter in phase 3 clinical trial, or it will successfully complete remaining clinical trials, and one day we can see this molecule in market in the form of immunosuppressive agent. So in conclusion, at the end, I would like to say that marine nature products are really having potential to treat many diseases, okay, because marine environment facilitates the biosynthesis of array of secondary metabolites which act as chemical weapons of marine organisms. In chemical ecology part, we have seen these molecules secreted by the organisms, whether by plants or animals or marine microorganisms, secreted by these marine organisms for their own purpose, for their defense, for communication in the form of infochemicals, or defense in the form of allelochemicals. So there are array of chemical weapons available with these animals. Now we have to systematically study the chemical ecology and try to develop these molecules for applications in biotechnology or in medicine. A significant number of marine compounds have shown potential therapeutic activities. Okay, the research work carried out so far highlights the importance of marine organisms as a new source of novel medicines. So these marine organisms are really useful. Definitely it's an unexplored area. Marine natural product research is unexplored. People are mostly in universities or colleges, people are working in these areas. Very few industries are involved. Of course, we have to name Pharmamer. Pharmamer is one industry which is specifically targeting molecules from marine environment. It's from Europe. So they are working in this area, and when I was showing clinical trial candidates or showing some market candidates, there were company names were also listed, and you could see many molecules were developed by this Pharmamed. So at the end, I would like to emphasize on the usefulness or potential of marine organisms in drug discovery process. Thank you very much.